Okay, welcome back, everyone. Welcome to session 3 of today's workshop. My name is Tannaz Ramezanli, and I'm senior pharmacologist within Office of Research and Standards at Office of Generic Drugs. And I'm leading the research related to developing cutaneous PK-based approaches using dermal sampling techniques at OGD. And now I would like to welcome our panelists for this session and invite them to share their videos as I introduce them. So we have Dr. Manfred Bodenlenz. He's a senior scientist at Yoon Research. He has been working with continuous sampling techniques such as DMD and DOFM for the past 22 years in clinical and preclinical PK/PD and bioequivalence studies. Next, we have Dr. Robert Lionberger. He is the Director of Office of Research and Standards in the Office of Generic Drugs, and he leads OGD's implementation of generic drug user fee amendments, science and research committees. We have Dr. Hiren Patel, who is the bioequivalence assessor at Office of Bioequivalence, and he's the lead for topical and transdermal drug products and the advanced techniques for demonstrating bioequivalence of these complex drug products within the Office of Bioequivalence. We have Dr. Raney, who was introduced before, and he's the Associate Director for Science at Office of Research and Standards, and he serves as a Chief Scientific Advisor for topical product bioequivalence issues of OGD. We have Dr. Elena Rantou. She's a lead mathematical statistician at Office of Biostatistics at FDA, and her work is mainly focused on assessing bioequivalence of topical dermatological drug products, characterizing outlier and detection of data anomalies. Next, we have Dr. Nilufer Tampal, who is the acting associate director for scientific quality in OGD's Office of Bioequivalence. And in this role, she develops strategies and oversees the implementation of data quality and the scientific integrity of bioequivalence data submitted in ANDAs. We have Dr. Eleftheria Tsakalozou, who is a senior pharmacologist at Division of Quantitative Methods and Modeling at Office of Research and Standards. And as part of her work, includes development and evaluation of quantitative methods and modeling tools to support bioequivalence assessment. We have also Dr. Miyoung Yoon, who currently serves as a team lead for quantitative clinical pharmacology team at Office of Research and Standards, leading the team's effort to conduct regulatory and research activities, leveraging clinical pharmacology tools and expertise. And last but not least, we have Dr. Jinhui Zhang, who is a chemist at Office of Pharmaceutical Quality, and his research at FDA focuses on using advanced mass spectroscopy and automation for clinical studies and the characterization of biotherapeutics and complex drug products. Okay, thank you very much. I would like to continue our discussion from the previous session about the design of the pilot study. So in the presentations that were given by me and Dr. Sinner and Dr. Kuzma, you saw examples of different types of pilot studies that we did to optimize the study design for the pivotal study. And I would like to ask your opinion about, again, we heard from several of our panels from last session that they would like to see they would think that pilot study would be needed prior to a pivotal study. But I would like to discuss what the design of the pilot study would look like. Do we need to use both test and reference product in the pilot study? Do we need to use an altered strength, maybe higher strength, and establish that the dose that we select for the pilot study and the design has adequate discrimination, or what controls we need to use in the pilot study? Maybe we can start with Sam.
Well, I'm happy to speak to that, but why don't we start with maybe with someone else, since I have been doing a lot of talking so far.
Sure. Okay. Anyone? Maybe Dr. Tsakalozou, do you want to chime in?
Yes, I'll be happy to share some thoughts. In my opinion, a pilot study or a method development study, however we would like to call it, is critical, mostly because of the unknowns when it comes to the cutaneous PK of many of the APIs and the products that we are studying. this is definitely a new area, an emerging area. So questions, as we saw, sampling duration, for instance, it may vary based on the API or the drug product. Also, as you mentioned, Tannaz, in your presentation, a dose to response or exposure relationship is not always straightforward. But when we want to compare reference to test, we want to make sure that we are at that range, dose range, where differences in the dose between the reference and the test can be detected with our methodology. Sample size is also another question. We do see high variability with this type of methodology, but we need to be able to have an understanding, a grasp of that variability before we conduct our pivotal study. So a pilot study, a method development study can give us some insight, provide with some observed clinical data in the population of interest, and then we can further use other tools, in silico tools, in vitro tools, to expand and make educated guesses and work on and make informed decisions on our pivotal study.
Okay, thank you very much. Anyone else, specifically from FDA folks, that would like to chime in?
Hi, Tannaz. I can chime in. I agree with Dr. Tsakalozou that it could be dependent on the product and API. And I also want to add that from a perspective of using in silico tools down the road to further support study design for the pivotal study down the road, there may be characterizing what's expected for the cutaneous PK disposition profiles from the pilot study would be really important. And it will help us to understand what portion of the cutaneous PK profile would be best representing the portion that would be sensitive to detect formulation differences. So that's something I wanted to add.
Thank you. So if no other comments related to this
I think
question,
Hiren had a comment too.
I just want
please
to
go.
add from the previous two panelists, I think Dr. Tsakalozou already discussed about the variability. And I think going back to one of your questions is which drug product to be utilized. So as we heard in the previous presentations, on what instances we are recommending, potentially the DMD or DOFM is when we have a Q3 similar product. So in my opinion, it would be a good idea to have at least the use of reference product. And since it's a different Q3, it's in an applicant's benefit if they utilize both taste and reference to understand what, Dr. Tsakalozou said, the variability. So ultimately it will help to power the pivotal study very nicely. So my preliminary thought is, the reference is must and taste. I mean, it depends, how they want to design the pilot study or so called the development or validation study. But yeah, the key aspect is I think reference product is something is very important to consider.
Okay, thank you.
I might just add, I think those are all great comments. I'll add very briefly that I agree that I think being able to demonstrate that the dose selected is appropriate is something that ultimately would need to be shown in the subjects. And therefore, I think that could constitute a pilot study where you'd be able to demonstrate that the selected dose is appropriate relative to being able to have a higher bioavailability differentiated if it did exist. I think it would be good to hear from Dr. Rantou as well in terms of what information would be needed to power the study from FDA's perspective. Is it necessary to have the test product in the pilot? But even more importantly, I know that Dr. Rantou has been doing some very interesting group in her work about adaptive study designs. And I think it'll be very interesting to discuss how much of the data in the pilot study, perhaps with the test and the reference product, can also then be leveraged into an adaptive study design for the pivotal study. But I see that a couple of other people have their hands up, which is great. So I'll step back for a moment.
Sure. Maybe if before we get into adaptive study design, we can hear from Dr. Lionberger and Dr. Zhang.
Thanks, Tannaz. So this is my comment on the pilot study is, this is, I think the reality is this is a new study design, right? It's not like, it's not like a normal PK study where, you really know how to do everything. And, so I think it's, certainly it's the applicant's decision to do a pilot study. I think it's sort of, not say silly of you to keep asking FDA, should we do pilot studies? I think that's your way to manage the risks on this. But I would also say that, if you think about this, for many products where we normally do bioequivalence in standard ways, like for example, PK studies, you can look at the label of the brand product. You can look at the freedom, and you can see like someone did a PK study on this drug, okay? But this is a new method. There's no brand or reference product that did a microdialysis or open flow OFM study on their drug to approve it as part of the bioavailability requirement. If I were on the last panel, then I could give Chinmay and OCP a hard time and saying, can you start doing start enforcing the bioavailability requirement against new drug applicants so they, so where the drug goes, right? So they generally don't do that, right? They look at systemic exposure as part of the bioavailability requirement rather than local exposure. So that's why I think you're in a very different situation here using this for bioequivalence, because the brand company, the brand product, there's nothing in their label about this. There's no literature on this. You don't really have even a good idea of what the value that you're going to get would be unless you do a pilot study, right? Maybe, hopefully our models are getting good enough that they can predict that. We have this new data on the local exposure. We're definitely going to use all the data that you heard generated in the first part of this talk to improve the models of skin absorption and distribution, right? So, you have a better chance of doing that. But really, I think, the fundamental thing is, without that pilot study, you really don't even know what level of exposure to expect. You can't just read the label and say, oh, Cmax is this. So I think in that, in that sense, I think that's the perspective on this, is that it is an emerging technology and you do have to do things a little bit differently than you normally would.
Thank you very much. And Dr. Zhang?
I just want to add a quick comment. The pilot study is also a great opportunity to understand the bioanalytical challenge. As Rob said, it's rather very new platform, right? And we solely depend on the measurement to make a decision for bioequivalence or not.
Okay.
That's pretty much what I have.
Yes, great point. Thank you. So now we can move to the discussion about adaptive study design and how we can leverage from the pilot study data to inform about the pivotal study, and whether we can use the subset of subjects that are used as part of a pilot study and include them as part of our BE study population.
Thank you. So first, let me add about something about the pilot study that so far the pilot study has been successfully used to evaluate variability, to estimate the sample size which is required to adequately power the pivotal study. But besides that, we have recent research that indicates that, similarly to the case of a clinical trial where an adaptive design can be used to decrease the number of subjects, to reach a decision with fewer subjects, to boost the power by saving resources, and also by controlling for the impact of the error rate, type 1 error rate. We can consider incorporating this methodology, the statistical methodology that we have, like, for example, the mixed scale average by equivalence approach, in an adaptive design in a case of a study like this, for example, in an IVPT study. So we can potentially leverage the data from the pilot study. One thing that we need to point out here is that we should have extra caution when we do that for various reasons. Mainly, as we heard in Dr. Ramezanli's talk, a pilot study is conducted to assist with the optimization of the bioequivalent study design. So we need to keep in mind that the pilot study's objective is different than that of the pivotal study. And there are some potential limitations and some things that we need to be extra cautious about. For example, blinding is something that we should worry about, because in an adaptive design, we should maintain the blinding. We should make sure that the design of the adaptive design is appropriate for a bioequivalent study. And also we should make sure that the measurements taken from a pilot study, it's realistic to incorporate them in the pivotal study.
Okay, so a follow-up question is, Dr. Rantou, if we have certain number of probe replicates in the pilot study, in order to use the adaptive design, we need to have the same replicate of probes for the pivotal study?
Yes. Mm-hmm.
Okay, thank you. I would like to discuss the use of redistribution probes and lateral diffusion probes as a control in the pilot and pivotal studies. So first of all, do you agree that we may need, based on the study that specifically I presented, it looked like if it even detect anything in these probes, it's mostly due to systemic redistribution rather than lateral diffusion. For systemic redistribution, even though the concentration we have found so far in or detected were minimal compared to what we detected at the application sites, do you think still we need to have these controls in our pilot, at least for the pilot studies?
So I can offer my thoughts briefly that I think in the pilot study, it is very important to have the redistribution probes to establish that the study conditions are such that you understand what the level of systemic exposure and systemic redistribution is. I'm not sure whether in then the pivotal study, whether it would also be necessary to do that. But I think that's something that we need to look at the data and think about a little bit more. I think Nilufer has also had her hand up.
I wanted to comment on, when I'm hearing about the pilot study, it sounds like different ideas are thrown out of about what a pilot study would be. And then if we are trying to kind of draw or use the data from the pilot study for the pivotal study in that adaptive design, we would expect, from a review perspective, that the conditions under which the pilot is done is the same as what the pivotal is done. So that's something I just wanted to bring out and make a comment here, because initially I'm hearing many different objectives of the pilot study. So, and maybe it's just in the name, how we are naming this. Are we talking about a part of it is as the validation to find the dose, the dose amount or the dose duration? We validate those things, and then in the pilot study, one of the objective could be that we want to assess the variability, and look at at the most TR ratio, but not really do a bioequivalence, not apply a bioequivalence criteria. And then maybe Elena, I'm not an expert here, but if in that case, maybe we're not even spending an alpha there, and then you kind of use that data and along with the pivotal for your two-stage design. So I just wanted to talk about that. Thank you.
I would like to comment on the redistribution probes. as Dr. Raney said, the degree of redistribution is very low. That's what we saw in the studies. And I would limit the use of extra redistribution probes to the pilot study only. As in the main study, we would like to use any probes and test sites for the main aim, which is test versus reference. So this question should be covered in the pilot study only.
Thank you for your input. Dr. Rantou, you were about to say something.
I'm not sure I understood the question, but the ideal situation is that we have the pilot study and the pivotal study under identical conditions. And in that situation, I guess we would have no problem with applying an adaptive design.
Okay. I see Dr. Tsakalozou wants to, okay, maybe we first go to Dr. Tampal if you want to address Dr. Rantou's comment.
No, this is not Dr. Rantou's comment, so I can hold.
Okay, Dr. Tsakalozou, please go ahead.
Yeah, just a quick comment that I agree with Elena. If the study design for the pilot study is very similar to what we expect for the pivotal study in terms of the dose administered, then having a redistribution probe is useful in the pilot study. However, if the dose is larger in the pivotal study, then we may need to employ additional tools to make sure that we don't have increased systemic exposure in the pivotal study. Therefore, whatever we concluded in the pilot is useful. And also we may use additional data to make an educated decision on whether a redistribution probe makes sense. So if we know that within the dose that we are administering, there's no exposure based on other studies, then maybe to decrease exposure, unnecessary exposure to healthy subjects, this is something that we need to decide to exclude. But also important for me would be the lateral diffusion probe. This again within the scope of the pilot study, assuming again that the dose is not significantly different between the pilot and the pivotal. So I agree with what Elena said. We need to closely consider to what extent the conclusions of the pivotal study can be extrapolated to the pivotal study, but they definitely, both the redistribution and the lateral diffusion probes within the scope of the pilot study answer important questions. Thank you.
Okay, thank you. And somebody has asked about whether they need to use dermal infusion in the pilot study. So the answer is no. The results that we presented regarding dermal infusion were just some mechanistic studies. We don't expect that you use the drug in the perfusate for the clinical studies. So Dr. Patel, please go ahead.
So I think as Dr. Tampal mentioned, there are a lot of objective, and we learned that, through either the research with the, what Dr. Kuzma presented or Dr. Senior presented. These are expensive studies, and the way development, validation, or pilot study are designed has multiple objectives. Are you trying to compare taste and reference? Are you trying to do different dose amount? You are using the ultra formulation. You are having lateral diffusion probe. You are having rate distribution probe. So when we say we can there's a potential if the study design is same for the pilot study into the pivotal study, but there are a lot of moving pieces. It is challenging how we can say that the study design is similar, because there might be chances that, in the pilot study, because it's the proof of concept, you are using 2 replicates, but when you go in a pivotal, that replicate number might increase. So it's just, I'm a little bit struggling to understand, how we can connect the pilot study into the pivotal study, or how we can utilize the data from the pilot study to pivotal study.
Anyone wants to answer Dr. Patel's questions?
Yeah, no, I think these are all excellent comments. I'll say very briefly that I think it's quite clear that I think Rob made this explicitly clear. This is not like you can look into a redacted NDA application and get information about your Cmax or where your time course is. Method development work will absolutely need to be done. The question, I think, becomes, does everything up to the pivotal study represent method development work, or would be submitted in the application and would justify all the conditions used? Or is there a specific pilot study, sort of like vasoconstrictor studies, we specifically ask for a pilot study? And there may be other things that are done before that pilot study happens. There are specific things that we want to see in that pilot study. And so I think, those are things that we don't need to work out in today's discussion, but these are really important considerations. And you're absolutely right. There are some things that would have to be identified, developed, validated before you even got to the conditions of the pilot study. And the way that at least I think about what a pilot study is, it's the exact conditions under which the pivotal study would be performed. Otherwise, being able to leverage the data set for an adaptive design just wouldn't be relevant. That's my only comment relating to Hiren's point, which I think is a really good one, an important distinction.
Thank you. So I would really like to move on to the next topic. Dr. Yoon, if you can go very briefly, so we can move on to the next topic.
Okay, so if we are using pilot study design, that's different from pivotal study design. I was going to comment that the lateral diffusion and the redistribution systemically, it is really will be dependent on the API. And rather than using the pilot study only, we have a lot of other methods to kind of do the educated guess on that part. Wanted to add that. Yeah.
Thank you. Okay, now let's discuss bioanalytical method validation and implementation challenges. So we heard from Dr. Sinner about some challenges associated with doing that and how it can be different for DOFM perfusate versus DMD. For example, microdialysis, our perfusate is a physiologic buffer and is not as complex as the perfusate we have for microdialysis. So I would like to hear from the panel about how we can overcome some of these challenges associated with bioanalytical method validation.
I can start.
Okay.
So generally speaking, the method validation still should follow FDA bioanalytical method validation guidance. However, validation is just a starting point, right? So, I mean, I think Sam and Rob emphasize again, it's the relative, rather very new platform. We know much less than the plasma PK and D study. So during the in-study method performance evaluation, that's very, very important. So, I mean, I just want to, in a minute, I'm going to paste a paper co-authored by our panelist, Dr. Manfred. I think his work on anti-clover, I think it's a good reference for us to look at the bioanalytical challenge and the bioanalytical method validation. So this paper focused on anti-clover, I mean, because anti-clover permission is pretty low. So they use the stable isotope labeled internal standard to track the matrix response. So I think it's a good reference point. And also, I'm going to paste in the chat one of our study using MEPT, though it's not DOFM or microdialysis, but it's also we point out several important points to keep in mind. Last but not least, to make sure whether the data makes sense and have you analyze to look at the raw data.
Thank you very much. Anyone else wants to chime in? Please go ahead, Dr. Tampal.
Yeah, Tannaz, so like in Dr. Sinner's presentation, he had one separate study just to collect the interstitial fluid. And I am my suggestion would be to consider, because these are expensive studies, right? And you're having the humans and just collecting the interstitial fluid. One thing we could consider is a surrogate. So a surrogate matrix, of course, when we use a surrogate matrix, we will have to do a good validation, a cross-validation with the actual matrix. So it's not that we don't have to collect that ISF, but we don't have to collect as much volume once we have done a cross-validation to show that whether I'm using a surrogate, a calibration curve in a surrogate matrix, or the one which has the diluted ISF, which is what the subject samples are in. You do a cross-validation, you show that the recoveries from the two calibration curves are similar. There is no matrix effects in terms of ion suppressions or enhancements. Once you do that, then you probably, for every analytical run, you don't have to make your calibrators in that same ISF, but you would still have your QCs, at least on a minimum, in the same matrix as your subject samples. But that will really help to cut down the cost of collecting just from the subjects, just for the sake of collecting additional fluid. So that was one of my comments that I wanted to make. Thank you.
So probably I can answer that. The regulation says that you need to validate your bioanalytical method in the relevant matrix, which in our case is dermal interstitial fluid. But this you can't just buy from the shelf in liters, as you can do with plasma and serum, but you have to do a study to collect it, which is very expensive. So yes, it's a good idea to use a surrogate matrix like a diluted serum, which as dermal interstitial fluid and is at least as complex in the composition as dermal interstitial fluid, and show that this is the same matrix which can be used validly for the bioanalytical establishment and validation. This would save a lot of time and costs, definitely, and this can be done.
Thank you. Any other comments? If not, we can move on to some of audience questions. So maybe this question is for Dr. Lionberger. Since these DOFM or DMD probes are considered a device, does conducting a B study with DOFM or DMD require a bio-IND?
Yeah, I mean, I would say I don't believe that they do, but I think there are some, our bio-IND regulations generally focus on the drug and the route, and this is not a new route for the drug or a new dose for the, a new dose for the drug. So, I would say that's a sense, but if you have a concern about a specific situation, certainly you can send in a control correspondence if you think that the general IND regulations apply to this. I would say, right, we have not, I think for our research studies, they've not been conducted, right, in humans in the US. So I think I can't give you that one, that experience there, that where we've conducted them, we haven't, seen these as different. But I remember when we first starting this, the probes aren't sort of, there's some, I would say some ambiguity around these studies as an investigation, especially when you're using investigational materials that haven't been approved for that and all of the different parts of that. So, I mean, you have to pay attention to the consult with your experts on the country that you're conducting the study in to comply with that. But, from our bio-IND regulations, most of this is the route of administration and the doses are appropriate. So from the drug product perspective, these don't seem to trigger the bio-IND submission requirements on that level.
Thank you very much. And the next question is for Dr. Raney. These drugs that would qualify for DMD or DFM study, they are applied in the studies that we have shown. We are applying them on healthy subjects with normal skin. How would this correlate with what happens in the disease or with the disease skin in patients?
Okay, that's a great question. So part of the reason that we have the framework of what Q3 same or Q3 similar, that ultimately what they both have in common is that all the Q3 attributes are well matched between the test and the reference product. And what that means is that the way that that product is going to interact with any use environment, whether the patient is shearing it at high shear rate, or whether they're applying it very slowly, whether it's in an environment of high humidity or low humidity, that whatever the disease state on which it's being applied and however that patient is applying it on the disease state, when all the Q3 attributes are matched, that significantly mitigates the risk that under any of those conditions that it's going to behave any differently. And so that's part of the reason why for products that are Q3 the same or Q3 similar, that we're able to conduct the study on normal intact skin, which is actually more discriminating than diseased skin. So under the most discriminating conditions, using a formulation that, because of its being Q3 matched throughout all Q3 parameters, should behave the same under any other conditions of use. That's part of the reason why those Q3 similar products combined with the DOFM study can sufficiently mitigate all the relevant failure modes for BE. However, for products that are Q3 different, you can't assume that. You haven't mitigated those risks. And that's part of the reason why for products that are Q3 different, it's not clear that there would be a different option other than a treatment of the actual disease state in patients.
Okay, thank you. And,
yeah, this is Bob. I mean, just a few comments. I mean, I think that's, you also have to think about what's the actual difference that you're looking at between the products, and is the microdialysis type study going to be appropriate for the difference that you're looking at? So, I don't want to say, like I said, we often do bioequivalent studies in healthy subjects because that's informative to what the products will do. And so if you're looking at, I want to understand what these excipients do to this, do to the skin, then you learn something from that. But certainly it's not the same condition. But so you have to think about that. But that's another aspect of where also I think as we build out our modeling approaches to both patients and healthy subjects, that really can be the place that really helps you bridge that and understand how to interpret the data if you think that there are significant difference. And and the other aspect of this is, what's the more sensitive discrimination of, did you change the formulation in a significant way? So certainly there may be more discussion around this, but the goal is a sort of an efficient bioequivalence approach where you have some difference in the formulation, and in vivo data will help you address that. It doesn't mean you have to do the bioequivalence study in the patient population.
That's a good point. Yeah. So I'll take back what I said, that even if it is Q3 different, that doesn't mean that DOFM wouldn't be, or DMD wouldn't be a suitable methodology. That's a really good point, Rob. Yeah.
Right, and you have to think about it may depend on what the difference in the, you may say, well, do these excipients affect the permeation across the skin, right? The DFM study would be a very good test of that. If you're worried about, does this irritate, a particular tissue, maybe that's not the right result, and you'd want to, so it may depend on the specific differences in the formulation as to what type of data you really need for the if you said, look, I have a formulation that's very different, not a formulation that's mostly similar, and I've also looked at the in vitro release and the characterization and the physical properties as well. That's part of the context of, is there a difference? And also, what the specific indication for the product is, right? And where its site of action is also very relevant to that discussion.
Thank you very much. So let's switch gears to data analysis of the pivotal BE study. So one thing we saw in the different drug products like acyclovir versus lidocaine, perlecaine versus diclofenac, is that the rate of drug permeation can be different, and it can be different depending on the formulation and also depending on the API. And when we did the study with acyclovir, we could not capture the terminal phase within the 36 hours. And the last time point was our Cmax. With the lidocaine, we had the Cmax within a few hours, and with diclofenac, we were able to modulate the study design by removing the formulation after a certain amount of time to be able to capture the terminal phase. So while we encourage the applicants to attempt to optimize the study design, but if the adequate portion of the PK profile is not captured within the 24-hour sampling, what is your recommendation? Would it be still appropriate to use that data to compare the PK, cutaneous PK of test and reference product? Or do we actually need to capture the Cmax within the sampling duration? Dr. Patel, I see you're unmuted, so please.
So probably I can start saying my thinking about this one and see if anybody else has to add anything. So, I mean, we also learned in today's presentation that, while developing the method or validating the method, there's an alternative strategy which can be utilized, like, wiping off the drug product to capture the elimination phase. And the reason I'm saying that one, as Dr. Kuzma presented, that when the drug was delivered through microdialysis probe, and we see the elimination from the dermal layer was completely different than actually from the drug product. So in my opinion, it is a good idea to capture the Cmax and certain region of the elimination phase to make sure that the test and reference product has equivalent absorption phase as well as the elimination phase. So this is my preliminary thinking on this.
Okay. Any other comments? Because sometimes the drug permeations is very slow, and we have flip-flop kinetics, and it may not be possible to capture the Cmax. What do we need to do in those situations?
Yeah, I mean, I think I think Tannaz makes a really good point, right? You can't, just because of some of the limitations on the time and the exposure, right? You can't really bring all of the concepts from PK studies over to this, right? Where you say, look, just sample the terminal region and estimate AUC infinity, right? Here, there's some physical impossibilities of doing that, and so, like, again, in some sense, this is a little bit of an emerging field, like what's the right way to think of interpreting the data from these type of studies, right? And, as you're in exploratory time, sometimes the modeling approaches can help and say, like, all right, here's what the drug would do, here's what the model predicts the drug would do over 7 hours, 7 days, right? But that's really not the sort of window you have into the product, right? Even with all of the improvements, there's only so much you can do. You have some control, right? You can decide, like, oh, I'll apply the drug for a certain amount of time and I'll sample, for a certain type of, will I take the drug off? There are ways I think you can optimize the design to answer that. But just to say that you'll have the same, that Cmax is the thing and the AUC is the thing, that just may not be feasible. And you maybe have to rethink how you think about the comparisons and what the right metrics for comparison are in these types of, in these types of study designs, which I think can be very informative to where's the drug at a certain time, right? But you'll never get, you may never get like the perfect, like complete coverage of absorption, and then I'll wait for it and watch it disappear from the skin, right? I don't know that that'll ever be, feasible unless the drug has a very, unless the residence time is just very low, right? That you can, that in that 1 day of sampling, you can see it come up and come down. I don't think that's the reality of many of the products. Some of them have different types of reservoirs. They remain there. They distribute then slowly into the rest of the body. But this is why you want to build up a good understanding of the drug distribution processes, and you can figure out then what's the most important thing to measure. And then again, remember why we're doing the BE study. It's to measure things about the in vivo performance that tell us about the product differences, right? So it's not that once the drug is in the body, then presumably it's going to be, if it came from the brand product or it came from the generic product, it's going to do the same thing. It's really that delivery aspects. And so what's the optimal study design to look at those different potential differences in the drug delivery? What's the critical aspect? We've done a lot of research, the group are looking at the aging of the formulations, right? How long does this formulation really stay in the skin when someone applies it? You want to capture all of those transformations. So, I think, again, this is still an emerging area. I would say we don't have the optimal design yet. And so these are things you have to think about in the context of a particular product.
Thank you very much. Dr. Tampal.
Yeah. So when I look at the acyclovir plot, right, from the microdermal diffusion, and like we also have the IVPT plots of the acyclovir, and we see like a profile versus for this other one, the microdermal diffusion, it's like, where is the Cmax? And I don't know where the Cmax is. I don't know where, so AUC is even further down, right? So couple of things, maybe is it like, is this particular product like a hydrophilic product versus a lipophilic product? And again, as Rob said, like, we still need to learn a little more about this methodology and where is it most suitable for. And then the other thing is like, what is there some sort of a modeling that we can be doing? Like, we have the AUC but we don't have the infinity. Can we kind of like do some modeling to get that rest of the information? So I think there's still some more that we need to learn from this technology, but it's a promising one, but we still need some more to learn a little more.
Thank you. Miyoung, please go ahead.
Yeah, I really like Dr. Tampal's comment about using simulation and modeling approaches. So I guess we will need a bit more information about what determines the cutaneous PK profiles and what exactly the permeation and the absorption part will determine, and what other physiological factors like a clearance from the skin or the metabolism is in the skin will determine the cutaneous PK profiles. So when we integrate those information, I think the cutaneous PK data would be a really good validation point when we use the modeling. And that way we can go further with the prediction of what's the outcome of after 24 hours, 48 hours, even if we have only segment of data at the time we sufficiently characterize the permeation part of the PK. Yeah.
Okay, thank you. And now I think let's go to some of audience questions, and we need to sum up in a few minutes. So this question may be for Sam. If we are measuring the drug concentration in the skin with these DMD and DOFM technologies, if the site of action is not in the dermis and it's in the epidermis or hair follicles, do you think still this method is applicable? And also, if the product is approved to be applied, let's say on the face, but we are measuring these drug concentration on the thighs, in the thigh area, do you have any concern related to that? Would it still applicable for products that are specifically approved for a different application site?
Yes. So my opinion is we're looking at rate and extent at or near the site of action. And the reality is that for most topical dermatological products, we're not really 100% sure where the site of action is. There might be multiple sites of action. In some cases, we may be more confident than others, but in many cases, there may in fact be more than one site of action. So I think certainly being able to measure the rate and extent at or near the site of action, it's all very proximal there. The skin is very thin. Is very reasonable to me. Certainly as reasonable as using blood levels to assess bioequivalence for a product that acts in the brain. As to the second aspect of it, I think this ties in a little bit with what Rob was talking about, is that the intention of the bioequivalence study is to consider what's the best study design to be able to detect whether there are differences between the test and the reference product, and whether the product is applied on the face, for which the skin is actually more permeable, compared to the thigh, for which it's less permeable. The thigh is actually more discriminating to differences. I think it's entirely appropriate to think about using the thigh as a very reasonable system in which to evaluate differences between a test and a reference product.
Thank you. And I think this will be our last question, that for drugs with long residence time in the skin, for which we cannot see Cmax in DOFM and DMD study, can we collect systemic PK samples in addition to DOFM and DMD samples to supplement our understanding of long life frame performance for the drug products? Any input?
So I think this is a really interesting idea. Maybe Rob can comment further on this. I think the question actually came in for Rob. But the idea of being able to supplement the data or supplement the modeling with in vivo systemic PK data, I think is a very interesting idea. And I think this is something we need to explore, maybe, to be able to.
Yeah, I mean, I think it's sort of, like, I think this would sort of fall in, like, sometimes we talk about in some of our other workshops, like model-based bioequivalence, right? How do you sort of put together, like, look, you have a drug where, look, you can detect, it's rare, you have to be able to detect the systemic PK after you've taken it off the skin, right? And you have, if you have different probes and a crossover, but maybe there's something where you can say, well, look, now I have an idea what the clearance of the drug itself is, right? Irrespective of where it came from. I fit that to a model by measuring the PK in the subjects. And then I plan, like, ahead of time to do something with the data that I collect from the microdialysis probes to develop my BE metric. Like, that's an example of a model-based BE approach, right? We would say, I measure this, I use this is how I analyze the data, you've pre-specified the approach, right? That's something that you could do that, might be valuable to sort of say, what's the right way to compare these products if I'm looking at the exposure at a particular site? I mean, you just have to do the homework ahead of time to figure out how to put those different pieces together.
Okay, thank you very much. And again, I encourage the industry, if you are interested in using these alternative BE approaches, you can engage with us through product development pre-NDA pathway at different stages of your product development, and we would work with you and give feedback on design of this the pilot study or the pivotal study. As you heard, some of these issues are product specific. And we don't know the answers unless we see some preliminary data. So I would highly encourage you to use this pathway to engage with FDA. So with that, I would like to thank you very much for your attention and engagement for this session. And thanks to all of our panelists for the wonderful discussions. Now I would like to hand it over to Dr. Priyanka Ghosh for workshop summation and closing remarks. Thank you very much.
Thank you so much, Tannaz.
Priyanka, I think you're muted.
Thank you. Thank you, Tannaz. Sorry, I thought I hit that button. Okay, thank you so much to every one of you who joined this workshop today and for sticking around with us till the end of the day. First and foremost, I would like to take this opportunity to thank our speakers and panelists and Tannaz and Sam for the wonderful sessions today. It was a lot of hard work to be able to summarize a decade worth of research and presented so that you can provide us with your feedback during this course of this workshop and in subsequent mechanisms for providing feedback. On behalf of our entire team, I would also like to thank all of you, all of the attendees, for the incredible engagement throughout the day and for sharing your recommendations and considerations using the chat function. We assure you that we will take all of those information and all of the discussion from today to design our research program for the upcoming years. With that, I would like to note that, as you may be already aware, the FY 2023 GDUFA research priorities in the topical space include development of efficient BE methods for topical drug products, which have differences, compositional differences relative to the reference standard. And a second aspect is improving comparative IVRT and in vivo cutaneous PK-based methodologies like the DOFM and DMD methodologies that you heard about, and resolve the practical challenges that we have with implementing these methodologies to support a demonstration of BE. And you can review all of our priorities within the GDUFA Regulatory Science and Research initiatives for fiscal year 2023, which is located on our website. The goal of our workshop today was to get your input and feedback on our research plans and enhance our understanding of the current challenges, which can further assist with the development of our research program in this space. As Dr. Luke and Raney mentioned at the beginning of the workshop today, the workshop was designed to discuss the current and prospective challenges with developing locally acting generic topical products. Our current recommendations for these drug products, specifically the characterization-based approaches, are summarized within our general draft guidances for industry on Q3, IVRT, and IBPT, as well as standardized and harmonized product-specific guidances that were designed specifically to facilitate your product development programs. As was mentioned earlier today, these guidances were published about 10 days ago, and the dockets for providing feedback to all of the guidances published in October are open till late December. So we encourage you to share your feedback and recommendations related to those documents as using the relevant dockets, and I have included the links in the chat for easy accessibility. Last but not the least, during today's presentation, we hope we were able to share with you what our current thinking is from both the industry and the agency perspective related to the next set of challenges for developing BE strategies for these topical products that may contain compositional differences related to the reference standard. And we also hope that the workshop was informative and allows you to consider these different options and share your feedback with the agency. Ultimately, when we have complex questions related to some of these locally acting drug products, as Dr. Tannaz mentioned, we encourage early engagement between industry and agency, specifically if you are using some of these innovative methods to facilitate the product development programs, and the pre-NDA program is a great pathway to be able to do that. With that, I would like to wrap up today's workshop by once again thanking the speakers, the panelists, the moderators, and all of you, as well as our entire team, both at the CRCG as well as the agency, who worked tirelessly to design and execute this workshop today. The slides and videos for this workshop will be available within approximately the next 3 weeks, and we will let all registrants know via email when they are available. Thank you so much for attending the workshop today, and hope you have a wonderful rest of the day. Thank you.